Um, okay, I'm sitting here with Hazel Screen. I think it's been a few years since I've came across your work on the interfascicular matrix within Tendon, which I had never heard of. Fascinating stuff. So why don't you tell everyone who you are?
All right, thank you, Jake. It's good to be here. Yeah, my name's Hazel Screen. I'm a mechanical engineer actually by training, but I've sort of always had an interest in bioengineering. So I've worked in that field, gosh, for the last 20 odd years. I did a PhD sort of back around 2000. Well, I really first started to come across Tendon, I guess. I had a real opportunity to take on what I thought was going to be a tissue engineering tendon project at the time, and I was going to understand something about the tissue. And it was an exciting opportunity to get a little bit more bio into my background work.
So this work with the interfascicular matrix,
Mhm.
how did that come about?
Um, well, I guess it almost did start when I started my PhD in a way. And I very naively went into my PhD saying, all right, if I want to tissue engineer tendon, you need to sort of look at the structure, see what's there. I want to look at the cells in the tissue. I want to understand the strains around that tissue, and look at how we recreate that if we start to build models. Um, and I very rapidly discovered I had absolutely no idea what the strains were around cells, nor did anybody else. Um, Nothing made a lot of sense. I started taking out bits of tissue and straining them and looking at the strains around the cell body. And the strains were much, much lower than what you were seeing at a tissue level. And when I actually sat and thought about it and put my proper engineering hat back on it, it makes sense. Tendons are fiber composite. It's collagen fibers. They're surrounded by a soft, gooey matrix. It's how we make fiber composites. If we try to make them in industry or whatever, tendon does it for us naturally. Fiber composites are clever materials in terms of how they strain. They don't just strain homogeneously. You wouldn't expect the strains all the way through to be the same. You'd expect this complicated environment where there's fibers and there's this surrounding tissue. So, I particularly got interested in the IFM because I got quite obsessed with just, well, how does a piece of tendon stretch? What's going on? If you just take it, what's happening all the way through these fiber composite at multiple levels, big, big structures surrounded by tissue, right the way down to individual collagen molecules. And I just did a simple experiment, really, taking whole tendons, and they were horse, I think, at the time, whole tendons, straining them to failure, and then taking the fascicles out of the tissue, straining them to failure. And the strains that the whole tendon could see were like twice as much as the failure strain for a fascicle. I was like, that just doesn't make sense. That's not possible. You can stretch the whole tissue twice as much. And I did lots of work, there's lots of stuff about how you grip the tissue, how you measure the strains, lots of stuff that could impact it. And, we took estimates on how it all could. Still, the whole tendon seemed to stretch too much. It didn't seem possible. So we started saying, well, okay, what else? How? Well, how about that gooey stuff between the fascicles that we started talking about that's holding them together? And therein lay a starting point for asking, what is it? What is the IFM?
Mhm. Sorry. um, I think I had found a paper, feel like it was in the 70s, where they had said tendon is basically an inert tissue. It's dead during
Mm.
life or something like that. Um, do you think that they're looking at a cadaver and things usually dry up and maybe they're just seeing the collagen? Is that kind of where you think that line of thought came from?
Mm.
Because like the IFM, does it have to be more of a fresh cadaver to see it, or does it sexually stay?
Yeah, it's there. You'll definitely see it more because it's more about, if you think about tendon is nearly all collagen. It's very dense, it's very thick collagen type 1, these really long aligned fibers. We're talking about a layer that's around them, and it's like a gel. If you imagine it like thick fibers with this sort of much softer, gooey gel surrounding them, as soon as your tissue gets dehydrated, yes, of course, you're going to see less IFM. But equally, if you look at the whole structure, if you look at a fiber composite material, you just sort of ignore the stuff. The fibers are there, aren't they? They're obvious. They're in front of you. Who cares about the stuff around them? I think naturally you just focus on those fibers that are in front of you.
Mhm. So you said the fiber composites like that we would make. What exactly? I'm not a engineer myself, so what kind of materials is this?
Well, okay, so you often have plastics, fiber, we have fiber reinforced, you have concrete, will be reinforced by steel fibers, for example. Plastics might be fibers surrounded by another type of plastic. But effectively in a fiber composite, you've got a fiber that's stiffer. In tendon, they're all aligned, aren't they, along the long axis, sort of aligned. Let's say they're aligned for now, all the way along the long axis of the tissue and surrounding them, holding them together, creating a full structure. You've got this soft, gooey matrix. So it is a little bit like a rope. A rope twists those fibers, doesn't it? But it's the same concept. You've got fibers all the way along, all effectively sort of bound together. And it's a great structure to build. One of the reasons we build rope like we do, because you know what happens if you just, damage the edge of a rope. The rope doesn't immediately fail because you just lose the ability for those few fibers to transfer load. You've still got some structural integrity. So you create these kind of materials so they're able to, to be a little bit more resistant to damage.
Mhm. Before we get on the specifics of the IFM, I had one day where I was like looking at the tendon hierarchy and the fascicles, and I was like, how many fascicles are there in a tendon?
Oh, that's a
Um,
good question.
so like if you took a horse, like a mature horse, how many? Just an
Oh,
estimate, how many?
right. I need to think about that a little bit. It is massively different. I'm gonna put off your question for a minute and waffle and see what I can come up with. Obviously, it depends on the size of the animal to start with. So actually, if you go to a rat Achilles tendon, there's effectively three from different muscle heads. It's right down at that size. An individual fascicle is of the order of point 0.4, half a, no, maybe zero point 0.4 of a millimeter in diameter. So you can start to get a sense. Um, you'll find that they're often grouped into groups of something like 20. It's like we take something like the horse SDFT is a massive, massive tendon. So the one you've asked me for the entire cross section, gosh, I don't know what's that. It's 5-6 centimeters wide, something like that. It's not round, it's thin, so it's about 2 centimeters thickness. So you've got them often grouped into sets of 20, 30. But you're going to have what, 10 of those groups, 20 of those groups. We should do some maths on it. We can. You can work that out, can't you? Let's
Yeah.
do it later.
Yeah, I did try to do some basic math when I was trying to figure it out. And I did see that with the rat. There was a nice photo of three fascicles of a rat
Yeah.
tendon, which was nice, but then they didn't have that for other animals.
Yeah.
So yeah, you said that horse SDFT, you've kind of compared that to the human Achilles tendon.
Mm.
What was the reason that we use that why do we use the horse to understand the IFM versus using like a rat or a mouse?
Okay. So I have used rats, Achilles a little bit as well. Um, done a little bit work in bovine tissue as well. There's a few reasons. Um, There's just the pragmatic reason of access to tissue, simple access to tissue. But you'd argue, of course, that rat and mouse is far easier to access. The real reason we've gone for the big tissues is because if you're well, certainly a mouse doesn't have multiple tendons feeding into the Achilles, so you don't see IFM in the same way. The entire tendon is roughly the thickness of one fascicle. So if we're looking at that structure, it's really hard to see. The horse is a lovely model. There's quite a lot of work that suggests it's pretty similar to the human Achilles tendon. The thing I particularly liked about it is that horses have a really, really good energy storage function. So if you're trying to look for subtle differences in how a structure is set up to enable function, it's quite good to look at extremes. So and in fact, you can see it. If you look at the work we've done in cow and then you look at the work we've done in horse, you see the same behaviors, but the horse is always the most extreme. So if you're trying to understand and explore, we wanted something that was really good energy store. I suspect we wanted kangaroo legs probably, but I was a bit short on supply on those. We can get equine tissue from an abattoir. It allows you to start to explore that in a model that's more representative of humans than if you start going into rodents.
Yeah, there was I think it was last summer they had I don't know if you were on that paper on the IFM, and they had like a sentence where they said it was the human and the horse will have a distinguished fascicle IFM, but the rodents, they don't have the distinction there. I think I might be a little bit off on that wording. Is that accurate?
Yeah, pretty much. I think it's you asked me how many fascicles there are in a tissue. The underlying bit that's interesting is fascicles seem to be roughly the same size in every species, roughly. There might be a little bit smaller, zero point 0.3 in a rat, but they're not that obviously different to a fascicle from a massive horse tendon or a human tendon, which is kind of interesting. But so if you can imagine, if you take, certainly we've done quite a lot with the rat Achilles because you can very clearly structurally see at least two different, probably three tendon heads coming together. And then you have that interface between them. So where the soleus and the gastroc are connecting, and that interface is effectively connected by a IFM equivalent layer. So that's quite a good model to work with. The mouse is so small. Um, you certainly have matrix between the fibrils within that tissue, but we're going down structural levels, we're going down. It's actually quite hard. Something you'd think was very simple, which is what is the hierarchical structure of tendon? If you really get into it, you're like, well, it does depend on the tendon. It varies. It's difficult.
Oh, okay. I've seen
Sorry.
I mean, I've seen a lot of tendon hierarchy photos. They're all, but I had never been like, yeah, questioning the accuracy of them all. Okay, so for people up to this point, I guess we've talked a lot about IFM fascicles. I think maybe for their understanding, well, now you've just questioned the tendon hierarchy. But anyways, the tendon hierarchy, you have the tendon, and then we have all these individual fascicles which have the strong collagen. And then kind of your work is that around the fascicles is you have this IFM type gel. So let's go a little bit further into the IFM structure function IFM. What have
Mhm.
you found so far?
Okay. So yeah, I think we have just at a global hierarchy level, perhaps to begin with, we have this idea. You go right down to the sort of individual collagen fibril. Yeah. So that's a nanoscale unit. I think Carl was talking about that here a while ago, wasn't he? That's a standard unit in collagen you see in all tissues. At that level, you have those units surrounded by quite a proteoglycan rich matrix, binding them together to make fibers. They're surrounded by a soft proteoglycan matrix. There's cells in the tissue there. They're bound together. They make fascicles. Fascicles are a useful unit for us to work with because you can extract them, you can hold them, you can see them. They're at that size scale, as we were saying, they're like 0.3, zero point 0.4 millimeters. So we're not talking, so when we talk about the IFM being between fascicles, there is a similar kind of soft proteoglycan matrix between the collagen all the way through the hierarchy. But I think the IFM is really interesting because it's a thicker layer. It's got a very clear, very defined cell population in it, and we'll get onto that. That's a lot of the other sort of aspects of that region that's key. So all the way through the hierarchy, though, it is a hydrated gel. You can think of it effectively like that. So it's full of proteoglycans. Proteoglycans are known for their ability to suck in, attract water, and really make a very hydrated gel. Um, there are a few collagen fibers in there, but when you look at tendon, it is unbelievably dense, isn't it? So the fascicles themselves are incredibly dense collagen. Between it, it's this soft gel with one or two fibers or a few fibers spread across between them. And we also find that most of the elastin in tendon is in that region. So, there's not a lot of elastin in tendon. Certainly not if you compare it with skin or arteries or anything like that. And, the collagen and the elastin make a kind of mesh network in those tissues so they can stretch and they can recover. You just you don't have that in tendon. You have this, 2% of the whole tissue volume really quite localized to the interfascicular matrix region.
Yeah, I've seen that with elastin. So the elastin and collagen, you think are like working together? Because I'm like collagen very strong, elastin has this capacity to
Mhm.
extend very far, and elastin has barely any. Well, not as much as like a skin material, but you think those two like kind of working together?
Um, well, I think everything in the tissue is working together. It needs to, in a way. You don't want your tendons too stretchy, do you? They're not going to do much use for you if they're like, skin or an artery. Your muscles wouldn't be doing anything. You'd lose all the energy of work. So you need something that's quite stiff. So your collagen's there. It's pretty stiff. It transfers force well. Um, what we see, and it is where the IFM became so interesting structurally, what's going on is you strain a piece of tissue that collagen stretches a little bit, but the stretch in the tendon, the ability for the tendon to stretch is enabled by the fascicle sliding. So one shears next to the other. Um, the proteoglycan gel will allow that. Nice, liquidy, lubricated. There's quite a lot of lubricant in there. Yeah, that's the lubricator in cartilage. Lubricated sliding. Um, but that needs to bounce back. So presumably the elastin is in there to allow recoil to ensure that you can get shearing, sliding between fascicles to allow the tendon to stretch and then to recover back to its original length.
So you said the proteoglycans within, I guess, within the fascicles around the fibrils, um, are they different than the proteoglycans in the IFM?
Probably. Yeah, it's all a little bit wave your arms around. We certainly know that there's a lot of decorin between fibrils. So that's where most of the decorin in tendon seems to be at that level. We find a lot less decorin in the interfascicular matrix. Lupus shows up quite heavily. That's a significant one. There's a range of other things like COMT as well, cartilage oligomer matrix protein. So yeah, they are definitely different. What I cannot tell you is if you go all the way through the hierarchy and define exactly what proteoglycans are at which level, what that looks like. Whether it matters or not, I also don't know.
Hmm. Yeah. Um, I guess we'll go further on that. Hopefully, when we get to pain. But so this distinction you've created with the energy storage and positional tendons. Could you kind of explain that?
Mm. Yeah, so when we did that first experiment, looking at the IFM and looking at how much the tendon stretches and then how much the fascicle stretch and seeing the significant difference between the two, that difference was massively more pronounced in the energy storing horse SDFT. It was like the tendon stretched twice as much as the fascicles. In the positional tendon, they were really quite a lot closer in terms of failure strain. You were like, well, that's kind of interesting, isn't it? There's something structurally, functionally different with how these tissues are straining or managing load. Um, so I think the more we've looked into it, the more you'll see that there is more elastin in that region, the recoil, the ability of the IFM. So we try to say, how do we actually mechanically test the IFM? That's quite hard to do because you can imagine you can stretch a whole tendon, you can stretch a fascicle. How do you get to this material between them? So we devised some tests where basically we could sort of take two fascicles and pull the opposite ends of them. So you could effectively shear the matrix between them and just see what it did. If you do that in energy storing tendons, it is far more fatigue resistant than it is in positional tendons. And we see these subtle differences in the composition. So there is actually slightly more decorin, I think, in the positional tendon IFM, but you see more elastin, you see more lubricant, all specific to the energy storing tendon IFM. The fascicles in those two tendon types were exactly the same. And that was sort of the next step in saying, okay, there's something in this. There's something really interesting in this interfascicular matrix. Collagen seems to be collagen, seems to be collagen. We're not seeing any difference in the mechanics of fascicles in all these different tissues. But the whole tendon properties are really different. And it seems to be coming from that interfascicular region. And then as we start to looking into it more, um, we started to find that, well, actually, the ability there's a really very active cell population specifically in the interfascicular matrix. The cells in fascicles don't seem to do a huge amount. They don't seem to turn over a lot of collagen. Although there's some interesting stuff. I think Carl was talking about, isn't there, the circadian rhythms and how they might have this pool of collagen. But the ability for interfascicular matrix cells to turn over the matrix seems to be like an order of magnitude better, at least. So you seem to have this active cell population that turn over the matrix more quickly, are more able to respond and change. So what are you, it's the chicken and egg. Is that responding to the load that the tissue is applied to? Is it something developmental? I don't know that. Well, we did do a development paper, but you start to see this tissue that sounds more responsive and able to change that bit of the matrix as its way to manipulate its mechanical properties. I said we did actually do a study of development to try to make sense. And again, the horse, you're able to do that. So we could do that from prenatal through to the first sort of couple of years after birth. And if you look at the tendons, sort of a prenatal stage, they are exactly the same. And then you can sort of slowly map them. It's interesting. You can sort of see the collagen starting to appear and delineate the interfascicular matrix in the energy storing tendon. So there's something in that initial loading. As soon as the horse first stands, you can start to see this development. So there's a clear link between the loading of the tissue and its formation in the first place to have this structural separation and specific structure to the IFM.
See that? That's a new paper or what?
Oh, I don't know. It was a couple of years ago.
Oh,
I have to look up where we published that. It'll come to me in a minute. I'll
yeah.
think about it.
Yeah. Okay. So to go back on the structure, we talk about I guess the cells within the fascicles, the tenocytes, tenocytes, whatever. Maybe like the not that active, but the ones in the IFM very active, turning over the matrix. What exactly are they turning over?
In any connective tissue, the cells are quite a small portion of the space. Yeah, the cells make the tissue. So you see that in any tissue we've got. So they are making those proteoglycans,
Mhm.
bits of collagen, probably the elastin. Very very slowly with elastin. There's this kind of. I don't think it's the case, but there's a sort of perceived knowledge that you've made all the elastin in your body you'll ever have, isn't there? By the time you're, I don't know, 10 or whatever it is, very young, and you just have no elastin turnover when you're old. I don't know if there's none. It's certainly very slow. But when we talk about the cells turning over the matrix, the matrix in all of our bodies constantly being renewed. It's like skin, isn't it? You know that because you lose skin, there's dust particles, you refresh it, you get new skin. All of your tissues are doing that, every tissue of your body, all the time in your life. We see that the rate at which that happens in the IFM is a lot faster than what happens um, in the fascicles themselves, and that's in health. That's just in normal, healthy, typical turnover.
Mhm. Yeah. I had on that circadian rhythm thing. It never
Mhm.
really. I was just walking one day and I had this speculation. I was like, because they've seen it in the rodents where it's like you have the little pool, 5% pool of consistently turning over. And then I was looking at IFM and like you have some collagen in there, and I was like, it would just make the most sense that the circadian collagen would be in the IFM. But is that just a speculation or?
It's speculation. It's not an unreasonable one, I guess. But yeah, it could be. Um, if we look, I was looking at EM images. Actually, we just got some really lovely EM images of the IFM, and you can certainly see fibers, and it's beautiful. They connect. So you've got your two fascicles, very aligned, very dense collagen, and these sort of actual fibers that bridge from one to the other. So there's definitely some collagen there. There's some elastin. It could well be. Equally, it is so sort of dense within that fibre, it might well be that there's somehow, I don't know, within that space already. I don't know. That one's beyond me, I'm afraid.
Yeah. Yeah, it was an interesting thought on my end, but I'm like, yeah, there's no way to confirm that. So when this energy storage and positional, you have that energy storage that gets the fascicle sliding, rotating, and positional is just kind of that I guess maybe linear strain, like they don't get that good sliding and rotating. When we look at young and old,
Mhm.
the old energy storage tendons, they start behaving more positional.
Yes, or less energy storing. Yes, I think again this was another just really interesting piece in that whole IFM focus. You look at aging, the collagen doesn't change. The fascicles have the same properties, the same behaviour. The IFM steadily gets stiffer. So if you start to look at fatigue testing, you really see that the change in fatigue behavior is reasonably specific to that IFM region, and it becomes stiffer. So in other words, it becomes harder to allow that shearing and sliding between fascicles. And the more we've sort of been thinking about it, I mean, with the horse example, we can go with the horse example. You could think the same thing with a human Achilles. It doesn't really matter. The tendon can't be expected to strain uniformly. It just doesn't make sense. For the horse, as it stretches, it goes round the fetlock joint. So you have to stretch it round a bend. If you stretch anything round a bend, the outside's got to stretch more than the inside, yeah? Or it can't do it. So immediately you've got to have that ability to have non-uniform strains and movement across the tissue. So of course, if you take that to the human Achilles, you know when you know you're using your soleus, you're using your gastroc differently with different movements, and if you're running, you've got quite a lot of gastroc effort. You have to allow those muscles to pull different parts of the tissue and for them to move independently, or you're going to start to get damage. And that effectively is our hypothesis. If that IFM becomes stiff, if you stop enabling that really nice, easy movement, sliding, non-uniformity of strains across the tissue, you start to load everything a bit too much. You start to get damage across that structure. And that's why with aging, as it gets stiff, you're much more prone to injury.
Mhm. So it's like that whole, if you have equal tendon strain, whole tendon strain when you're young versus old, you're thinking that you're getting more strain on the fascicles, fibrils when you're older.
Yeah, yeah,
Yeah.
I would imagine so, yeah, because it's
Yeah.
less well, and it's just not well distributed. So if you are using your, I don't know, your soleus more than the rest of your tendon when you're loading, you're no longer able to redistribute that strain through the Achilles as well, and then you'll start to get injured.
So IFM gets stiffer. Um, what's going on structurally when it gets stiffer inside the IFM? Is it getting less volume? Is it getting like a change with its proteoglycans or what?
So what can we see? It seems to be a bit less hydrated. We do see some changes in composition, and I can't remember all of them off the top of my head. There's a long list of, audited the proteins that were in the IFM when they were young and when it was old. And there were some changes. So you can see some proteins go up a bit, some go down a little bit. Um, Structurally, none of the techniques that I've looked at yet show a really obvious structural change, but that's quite hard to see. You're relying on um, well, you could do it with SEM. I don't know if we've done a young old with SEM yet, but normally you do it with like histology and look at slides, and the sort of resolution you'll get with that, I don't think will show you a clear and obvious structural change, but we do see changes in the protein makeup. And the cells themselves seem to start to change phenotype a little bit. So again, you see slight differences in the genes they express, and therefore the matrix they turn over and how they behave.
Mhm. Um, do you see a volume decrease? That's more like it's less hydrated. So would that just?
Yeah. Yeah, it's a really, really hard question because of hydration. It's one of the big things that always worries me about describing, when you talk about tendon, you talk about mechanical properties. We typically, the sort of ethos in engineering, isn't it? You don't talk about a failure force, you talk about a failure stress because you're normalizing to the cross-sectional area of the tissue. That makes absolutely perfect sense for a piece of steel because if the piece of steel is bigger, you'd expect a higher failure force. It should be a beautiful linear relationship between the two. It's actually a very difficult relationship with fiber composites, particularly hydrated fiber composites like tendon, where it depends quite how hydrated the tissue is. So volume measures, how big, it's really hard. You think about the processing you have to do to try to look at it. You have to dehydrate it, and then you don't quite know. So it's a very, very difficult question actually to answer.
Mhm. Yeah, I uh, I had that with one of my friends track guy was talking about having a thicker tendon, how it's better, and I'm like, well, probably, but also when you get tendinopathy, it will get very thick, and that's not a good tendon.
It might be really, really bad, if
Yeah,
that's yeah, if you've got sort of swelling in there, I don't know, too much water imbibed, it's yeah, hard
yeah,
to know.
yeah. Okay, so that's the next part I want to go on is this healthy tendon versus painful tendon. What kind of changes? I remember you had that paper with Jill Cook a few years
Mhm.
ago, kind of looking at the tendinopathy continuum and saying, what was it called? Did we miss the first something about the IFM? But yeah, go ahead.
Yeah. Oh God, yeah, I think it was did we miss the first step, I think she talks about. I think, it's fascinating some of the stuff she was looking at and this kind of, idea of, you start to kick off reactive and how do you manage that process. I think really, well, we did a series of studies in, explants, and we've done one recently actually in an in vivo rat model. As I said, we will use the rat Achilles model as well. We said, let's try and look very early on that if you just cyclically load the tendon, what's happening? What response do you see? So we did it with I think coarse explants. I think we've done some bovine explants and the rat in vivo. And in each of these cases, if you just apply, I don't know, 10,000 cycles or something like this. So it's a living explant of tissue, or it said it's in vivo in the rat. You stop there and you look at the response in the tissue. You can look at, some of the early kind of markers of load. So one thing you can look at, and we looked at in the rat in vivo, is alpha smooth muscle actin. That's something a cell expresses if it's experienced mechanical strain. So it's its first cue that it's being loaded. And then you can start to look at some of the inflammatory markers, the degradatory markers, things that cells might start kicking out. It's only gene expression level, so it doesn't mean it's going on, but the sorts of genes that they might kick out to say, let's start to, start a low key inflammatory response. Let's start to degrade the matrix around the tissue. All of that was localized to the IFM. So all of that data seems to be implying that that very first kind of response to overload was quite local to those cells that were responding with kicking off IL-6 and matrix metalloproteinases to start to break down. And that's what you'd expect if there was just a very low level of damage to the fibers. It's not a problem. It's almost normal everyday life. A tissue will repair it. But for a tissue to repair, the way it does it is give you a slight inflammatory response. It starts to degrade the old matrix and then starts to kick out a fresh new matrix. So it starts to show that that might be the first thing that happens. Um, I think that paper then went on to say that, there's a point where that's goes from being the healthy standard response to use to being too much. And it's like there's a tipping point, and that's, Jill's reactive tendinopathy discussion and back to that kind of process. But I think the bit we were talking about is where does that process start? And it's sort of a little bit against what you were originally seeing as a perceived wisdom. Because, of course, if you look at a tendon down the line, you see this spaghetti, don't you? You see the fibers all over the place. It's in a mess. You're looking at something very downstream from that really initial response of the cells.
And that's so I the reactive tendinopathy. I mean, I've had it many times. I had it in my Achilles once, and it was like swollen, red hot, very sensitive. Um, a lot of that because the IFM has so many more cells, like the pain, were you thinking with the pain response? Like pain is always very difficult to talk about, but it's like I know we have this idea of nerve ingrowth into the tendon, which it shouldn't be in there. Maybe that's causing pain. Maybe
Mhm.
it's like chemical signals. Initially, in that reactive stage, you're thinking a lot of that is probably happening for IFM.
I think it probably is. I am not, certainly not a pain expert. I know that when you do get ingrowth, it tends to ingrow within that interfascicular region. It's where the blood vessels are. If you look in a natural, healthy piece of tissue, that was that traditional view that it was completely avascular. It's not. The blood vessels run through the IFM, though. It's a bit more spacey, isn't it? There's space for this sort of stuff to happen. So my assumption, and it is an assumption, I don't know, is that you'll see that process starting in that region and that ability for the cells to respond.
Mhm. there was so later, I think it was after that study, Lauren Pringles, I've talked with him a few times. He had a paper on, I think it was on pressure within tendon, but he had these
Mhm.
diagrams of the first stage of reactive when the IFM kind of swells up, but then he had those individual fascicles, and then it was like the fascicles are opening up, and so like you no longer have this distinction of, and I think you guys actually had that in your paper, how you had that photo of the nice two fascicles going, I guess maybe they were going opposite ways, and then you had the IFM within, but then when you overload it, it's like this, they're mixed together.
Mm.
So when you have that like acute overload, or maybe if you continue it on, is there no longer this clear distinction fascicle IFM?
And I think that's where we've not tracked it all the way, but that's certainly what you see when I'm talking about this very early reactive response. There is no structural change. Structurally, the tissue looks perfect, exactly the same as it was before you started. This is a cellular response. Um, what that cellular response will then do is start to kick off a cascade of churning over the matrix, changing the response. And I think there's a point there that if you just give it time and it recovers, it will just settle down. Your structure will never change. Everything's happy. If you continue pushing that and loading and overloading, you'll start to see structural damage. What I have not seen yet, but my hypothesis would be that structural damage would start in the IFM. As you said, it would open up and then steadily your cells across the tendon will respond to breaking down and giving you this mush matrix. And then you have no clear, certainly at an endpoint, there is no clear, distinct IFM. And at that point, you can say, well, once you haven't got that, you have no nice, clear ability for that tendon to strain properly, to do its shearing, to be able to load uniformly. And that's when you've got a tendon that is going to be difficult to manage and to look at structurally how do you get that back to functioning effectively.
Yeah, I've seen some photos of the cross section of the horse SDFT where like they have those clear fascicles, and then there was one study that had an injured one where it's all just this. Yeah, there's no distinction anymore. Do you think that if it gets to that point, can it recover and go back to the normal looking tendon? Or obviously depends on the extent of the injury, but I'm thinking if it's like a short term acute thing, do you think it can recover and go back to a normal function?
I think you can probably get back. So I don't know. I'm not sure if it depends definitely how bad the damage is. I reckon that to get that structure back to perfect, it's quite an ask, isn't it? And I think if you look at the ultrasounds of people post injury for long enough, I haven't seen a great deal of tendons that have gone back to looking structurally perfect after going through an injury cycle. We don't know enough about that. I feel you should be able to recover a degree of functionality. And I think one of the things we're quite interested in at the moment and trying to develop is ways that we can um, sort of use ultrasound to measure sliding, really, to actually be able to detect how that tendon shearing and sliding, to take people, look at how it changes with injury, and then to look if we can start to take people through rehab programs, if we can monitor that, and if there are certain types of exercises or approaches that might help enable sliding and structurally. I'm not convinced it's going to look beautiful, but if structurally you change it to be able to shear somehow, then you've recovered the function, which is probably more important than what it looks like.
Yeah. The I guess we could talk human or horse when they rupture that Achilles or SDFT rupture,
Mm.
and then they I guess humans can go non-surgical. I don't know if they ever do that in horses. But is it never? I've just always speculated. I had a lot of friends. I'm thinking like that is never going back to a good sliding and gliding of fascicles. It's just this scar, and you're gonna have to deal with that. Is that kind of what you're thinking? Is like it's never going back?
Can't see it ever going back. Whether you can do some, yeah, it's never going fully back. I wonder how much, movement and shear, if you can get any shear planes, if you need them, if you can get the rest of the tendon to manage. Because you imagine you've got the whole length and you might have a region in the middle that's stiff. and it's not really doing anything, it's never going to be good like that. But if you can get the rest of the tissue to function well around it, you probably get reasonable function out of it.
Mhm. Yeah. Um, let's go away from pain for a second.
Mm.
This podcast is going all over the place because this is
Yeah.
how it needs to go. But the we have this idea with tendons that, so yeah, we'll take a healthy tendon because it's unhealthy. The function is, I guess, who knows how it's working, but viscoelasticity. So
Mm.
I kind of got on this personally when I had jumper's knee and I started doing isometrics, like holding positions. And then there was this speculation of like the collagen is certain things are happening at the collagen level. And I was behind them for a while. And then I was like, I don't think that's happening at all. What, what do you think is happening when we talk about viscoelasticity with tendons? So if you're doing something where the tendon is holding a position for a long
Mm.
period of time versus the tendon is quickly storing and releasing energy, yeah, what do you think's happening?
Oh, yeah, that's difficult, isn't it? Okay. I can answer that in different levels and different ways. Let's just take isolated bits of tissue, you know. 'Cause I think yeah, that strikes me as the harder bit to do to begin with, because I think at a whole body level, you've got the muscle, you've got everything else, it's bound to be involved. If you take an isolated piece of tissue, um, and you strain it, let's just say you strain it and you hold it under a set amount of strain, we know that that force will just steadily drop off. In theory, it will, drop off to nothing if you wait long enough. Um, we know that when you started, the collagen was under, each collagen fiber was under a certain amount of strain. It was stretched. We know that they are pulling back. It's got to be shearing, sliding within the matrix that's enabling that. Collagen's reasonably stiff, doesn't want to be stretched. You hold it, the collagen will return to its original length because the fibers will shuffle, they'll move, the matrix around them will enable that. So I think if you do that at a whole tendon level, you're going to see that same sort of behavior. The interfascicular matrix is going to allow that reorganization to basically take the load off the tissue and return it to each collagen fiber to an unstrained state where the whole tendon is longer than it was when you started. Okay, so when you do that in the body, though, you've got a muscle, you've got that coming into the tendon, connecting, to different fibers separately. Um, Muscle in its own right is viscoelastic. It will be able to extend a little bit. I think you'll transfer that all the way along that entire unit, and you'll get effectively a little bit of reshuffling of each of the structures to slowly dissipate the amount of strain and start to bring things back to their original length. Because any material doesn't, it requires energy to be strained. You have to put energy in to stretch it, and you have to, if you're holding it at that strain rate, if it can release that and go back to its original length, it will. So that's what you're seeing. Your whole unit, you're holding, you put in energy, you're holding it strained, and it will try to dissipate that by moving around water, moving around molecules, and letting the collagen structures relax. So I think that's probably what's happening. Now, what did you want me to answer on this one? We were talking about fast movements, slow movements, I think, didn't you? What's happening
Yeah,
this
but maybe could we stay on the
Yeah,
slow
go
for a
on.
bit? Because I had
Yeah.
never I've been searching for a long time to know what's happening in that holding a strain when you hold the position or the tendon is held. So you're saying that whatever, say it's like I don't know, 4% strain that it's like set at, we're gonna hold it at this length and it's gonna like stay there. You're thinking that the collagen within is already at its point when you start it, and then it actually crimps as you hold it long enough. It goes back to a crimp state.
Cover back, yeah. You'll hold the whole tendon, and yeah, you remember this. An isolated bit of tissue doesn't have the body responses and everything else. It's less ability for fluid influx, less ability for your muscle to respond and do things. But if you just take a piece of tissue and you strain that piece of tissue to 4%, you'll strain the collagen less than 4%. I don't know, 2 or something.
Mhm.
Yeah, you'll strain the fibers themselves, and then you'll get shearing in the IFM. You'll strain it, and then you just hold it there, and that collagen will go back down to its original length, And the IFM will allow the shearing and sliding, and it will redistribute until it's back to having the minimal force state for that tissue. So the minimum amount of tension for the individual components.
Um, is there a is it is there a time frame for that to happen?
Depends.
Yeah.
um, depends on the tissue, depends on lots of things. If we did say so we call that a stress relaxation test if you do it in a piece of isolated tissue. A lot of the stress relaxation tests I did on just fascicles, we'd do, say, a 5-minute test. And over 5 minutes, if you applied 8% strain to the fascicle, the force had dropped by 50% by the end of the 5 minutes. And it sort of plateaus. So you see a very rapid early drop, and then it sort of plateaus out and it will stay there for a while. But gradually, it will keel off. But over the first minute, you imagine get a lot of water movement, a lot of other, structural changes in that tissue. You see a relaxing of the load literally over a minute very quickly, but then that peters out. So you don't dissipate all of that strain across all of the tissue instantaneously, but you redistribute it certainly over a 1-minute time scale to begin with.
Hmm. And that water, I guess you maybe have to look at the in a human tendon. It's like the
Mm.
water is coming out of the tendon.
Oh, that's maybe.
Uh-huh.
We did actually, so yes and no. So I think at a whole tendon level, I think some of it does come out. If you look at the thickness of the tendon and you hold it in a stress relaxation state, the whole tendon gets thinner. But we did actually do some sync. Oh my god, this was years ago. I'm trying to remember the studies. Back in about 2005, I did some work with Marjorie Gupta. He's a collagen sacs expert, and we took some tendon fascicles to the synchrotron, and we did stress relaxation tests with them. And at a molecular level, they seem to imbibe water. They seem to get fatter. It was really quite strange. So the entire fascicle would get hugely thinner. You'd apply load and you could see it started at, it started at a diameter of zero point 0.4 of a mil, whatever we said a fascicle was, and it was half. So it really reduced in thickness over the time of this test of 5 minutes. But within that structure, there was very clearly quite interesting movement of water. Some appeared to be being kicked out of the whole tissue. Some appeared to be going in at the collagen molecule level as well.
Yeah. Okay.
Yeah, ouch. I know. Yeah, it made my head hurt. But I think if we do this at a whole tissue level, we're
Uh,
probably seeing a loss of water out of the tissue.
it would make me think if the collagen is actually crimping, would
Hmm.
it then make sense that water would be going back towards that?
Yeah. So when you say, I mean, yeah, you're when you stretch the fascicle, you'll effectively pull out that crimp. It will, recover back down. Water could move around within that. But I think, yeah, globally on a whole, I expect if we look at a fascicle, the overall reduction in its diameter suggests that you're kicking water out.
Yeah, that's okay. I didn't the that way of thinking. I'm gonna need a while to process that. But that's like so like when we think of a store and release thing, like a fast thing, and say the tendon straining 10%, it's like the whole tendon 10%, maybe the fascicle 8, fibril 6, 4. Like so, you would agree with that. But then if we're holding a position, it's like you have the set. If it's a 10% strain, that it's actually getting less at the over time. It's getting less at the fascicle fibril collagen level.
Mm. Yes.
Okay. And
Yeah.
that's what stress relaxation is.
And yeah,
Okay.
yeah, effectively. All stress relaxation is a structure redistributing the strain that it's under to be in its sort of minimal energy state. It doesn't want to be stretched, so it's
Mm.
trying to, release that stretch. Think about it as sort of, wiggling, getting comfortable, moving things around to
Okay.
release that strain on it.
I'm only like 5 years into looking into tendons, and like that's one thing I had missed, and it's probably my own incompetence in like the engineering world of how materials work. The oh, where was I gonna go with this? I had somewhere I was gonna go with that. It's gonna come back to me, but can we go on now to the I said visco elasticity, talked about the viscous property there. Oh, what I want to say is tendon creep. How does tendon creep fit into all of this? The tendon getting longer as you hold the position.
So to creep a tendon, you have to hold the force. So what you're doing then is you're putting a constant force on it, and it gets longer and longer and longer. And if you do that, not in the body, one way people have done it is, say you just hang a tendon. So you grip one end and you hang a great big weight off the other end. If you hang that weight off the other end of the tendon for long enough, if it's heavy enough, the tendon will creep until it snaps. Yeah, I mean, some tendons, some of them really, obviously, the Achilles is incredibly strong, as is the equinus, the FT. It would be one hell of a weight to get it to snap. But if you imagine that on a smaller scale with a smaller tendon, it will creep, creep, creep until it snaps. And again, it's how does it manage? It's got that constant force constantly pulling at it. So you stretch the fibers, but now you're looking at how they'll be rearranging, the water's moving, the materials are time dependent. Slowly it pulls apart. And you can see if you do that, I've not really done a static creep test, but I would do a cyclic one. It speeds it up. Imagine I'm talking about slowly structural reordering. If you cyclically do that, you speed up that whole creep process. So you keep cycling to a load, keep pulling it to the same amount of load, and it gets longer and longer and longer. And you actually can watch it unravel. So if you do that in a test on a machine, you can see a ping, one fiber goes and it twists, it unravels, and then slowly the whole structure untwists and pulls apart. And you get these huge, long bits of sort of pulling apart collagen fiber as it just completely fails.
Mhm. Yeah. Okay. When we look at a fast movement,
Mhm.
so this is not, I guess you do get the tendon creep if it's I think they've seen that in marathon runners, some of them as they
Mhm.
get longer in the race, that the Achilles gets a bit longer. Yeah, what's happening in fast when you move a tendon in a fast manner?
Well, you're not, yeah, you're just not giving time for time dependent effects to occur. So, that's the faster you strain a piece of tissue, the stiffer it is. And the less time you allow for shearing, sliding, water movement, anything else. Um, so it's quite interesting from an IFM perspective, obviously, as well. If you do very fast movements, that IFM has to allow that shearing and sliding. It can't be as just a slow, viscous, time dependent response. It needs to be able to allow shearing quite rapidly. Um, yeah, does that answer it?
Yeah, so that's about it because they're very similar then.
What did you mean in terms of how tendon stretches? Yeah, physically,
Stretching,
what's
yeah, stretching
happening?
slow and fast, like very similar. It's just that fast you don't get the time dependent things.
Yeah, I mean, what's physically happening is the same thing. You're just allowing a lot less time for it, and by allowing a lot less time, you're allowing less time for water to move. You'd expect the whole tissue to behave more stiffly. I'd expect, you see the collagen behaving more stiffly. You see the IFM behaving stiffer, allowing a little bit less shear. So it's quite an interesting thing. And I don't know the answers to this at all, but it's something that interests me to ask, um, how you sort of think about exercises, rehabilitation, supporting, managing for patients, asking the importance of, doing sort of plyometrics, high speed exercise. I see it's got to be crucial there for the function, but how you get that right, I don't know. So what you're doing and how you gradate that kind of process of rehab for, whether you need a period to be stretching to allow that, try to encourage that sliding to recover, to happen. Maybe then as you start to get better, you need sort of plyometrics, rapid loading to allow that response. I don't know the answers, but somehow in my head it seems to make sense to me that early in injury you want to start encouraging sliding, and that requires a little bit of holding and allowing some time for these shearing movements, and then you need to gradually ramp that up.
Yeah, it's just a big guess on
It's
everyone's
a
part.
massive yes,
Yeah.
yeah.
Yeah. So when we have those going to the Achilles, they have that research on the three subtendons and how they have that twist.
Mm.
With the fascicles, is there also a twist happening? Because so the fascicles go from the muscle to the bone. Are they all going muscle to bone, or some of them actually
Depends who you are.
originating? Yeah, yeah.
Um, I don't think they can be. Nobody can properly tell. That sounds a bit bonkers, but it's actually unbelievably difficult to tell how long they are because they are very long. So in, fiber composite terms, how long a fiber is quite important because you imagine if it's very, very short, it doesn't have much surface area to transfer force between it and the surrounding matrix. So, there's a sort of theory behind how long a fiber has to be to transfer force and how to function. They're clearly long. And in that sort of fiber composite language, they are long enough to almost count as continuous. I don't think they can run from the muscle to the bone, though. I mean, if you look at, so take the Achilles, it starts one shape and it ends another. It's got different thicknesses from the top to the bottom. The shape changes. Um, I don't know how it could happen. And equally, if they are connected top to bottom, the shearing could simply be to allow some to be loaded more than others. But my feeling is that they are just very long but discontinuous, and that matrix between them, which is actually quite strong. If you look at its mechanics, it's perfectly strong enough to withstand forces and to hold the tissue together and allow that shearing sliding behavior to happen. So I don't think they connect top to bottom. They are in groups. Those groups sort of follow around as you've got um, the sort of rotation in the tendon. Interestingly, the actual fascicles themselves are helical as well, though, in energy storing tendons. And the one difference we found in the actual fascicles of an energy storing in a positional tendon. So we did that in the horse. And we took out fascicles and we drew like little grids on them. With stains under a microscope, and we strained the samples to see how the grids deformed. And you could basically see if you strained a positional fascicle that the fibrils within the fascicle was sliding, and the grid was kind of deforming, so you could see them moving different ways. When you strained the fascicle from an energy store, the entire thing rotated. So it was like you've got these little helical springs of fascicles within the tissue as well.
Yeah, I've kind of people have that term, the fractal of like the micro level will mirror the macro. So do you think, at least with the Achilles, you get that twist and then the fascicles are twisting? Are the fibrils also twisting? And then the collagen definitely is because it has a triple helix, but
Yeah, maybe. I don't know. I'm not quite sure how you'd look at that. It'd be quite difficult to sort of, you're down at electron microscopy sort of level to try to see that. And you've got these incredibly long and incredibly thin fibers. And that's why it's so hard, because to get enough of a magnification to see the diameter of a fibril really well, and you can see very little of its length. So that ability to explore what's happening at that level is quite challenging.
mm,
Um, but yeah, you certainly see this repeat of this kind of helix, so it wouldn't be shocking, would it?
yeah. The there was yeah, I remember that. I think it was that paper. The fibrils are continuity through the whole
Mhm.
thing, and yeah. But then there was some research on them breaking and then branching off and connecting to one another to like maintain
Yeah.
integrity. Do you think something similar happens in fascicles, or they're not? I guess they would adhere through the IFM, right? And there wouldn't be
Yeah,
like a yeah.
I've certainly never seen branching fascicles, but you can see. I mean, if you look at histology or EM pictures of the interfascicular matrix, you can see it bridging. So it is connecting those two fascicles together at that level. So yeah, it is. It's an ongoing. We don't know how long the fibrils are, or the fascicles are, or any of those structures in tendon, and it probably depends on what the tissue is. Because if you're talking about a fibril that covers the entire length of a mouse tissue, well, that's 5 millimeters compared to a fibril that covers the entire length of an equine tendon. Well, that's like, I don't know, half a meter. It's a very, very different sort of scale that we're talking about. And it is one of the endless things we hear, tendon is tendon is tendon. Well, No, that's difficult. They're hugely different.
Do you got a few more minutes?
Yeah.
Okay, perfect. I put it for an hour, but I got a few more. So all that research on, actually there probably was some on patellar and IFM, but the Achilles and the SDFT and the energy storage and release. What have you in human patellar tendon? Do you
Mhm.
think the same things apply to a lesser extent, or what are you thinking?
Yeah, maybe. I don't know a lot about the patella. Um, it's a much shorter tendon, obviously. It's not got this kind of multiple um, muscles directly attached because you go through the patella. Obviously, you're not coming straight from the gastroc, but you have got loads across, uneven, non-distributed loads, haven't you, across different parts of the leg, depending on how you use it. So probably. There was a couple of studies. I think Laura Slane did one, and the group there in Nervan were looking at trying to use some ultrasound imaging to try to look at how uniform strains were across the patella during use, and started to pick up again indications of non-uniform strain distribution when you use the patella. So it seems yes.
Hmm. Yeah, yeah, yeah. I've had that thought recently with the. I mean, you were saying earlier with when you hold that, like with patellar tendon, like I found so much benefit with holding isometrics or doing slow, heavy strength, and recently had that speculation of like maybe it's when I'm doing the very fast movements that are provocative for the tendon, it's overloading certain areas like
Mm.
of the tendon. But then when you get and do like a slow heavy move, you get this reorientation. Maybe you're getting load in different areas that you otherwise could not. But then I looked at I've read a lot of these horse tendon books and like look at a lot of horse tendon research, and I'm like, they're not doing isometrics, or they're not lifting weights for the rehab. So I'm like, how do they get the distribution of load to different areas? Like why are they able to do it? And then for us, it's very hard for us to rehab our tendons with just plyometric things. Like we always need to add in the lifting type movements. I don't know if you have any insight there.
Oh dear, I do know that they're a pain to work with. I said never work with animals and children, they say, isn't it? But yeah, we'll do a lot of our studies in humans to set it up to be easier to prep for horses. It feels very backwards, doesn't it, in how you run things. But yeah, you can't tell a horse to do an exercise at a certain rate and respond, and load its tendons slowly so we can see what's going on. Um, I don't know, they're heavy though. And if you think about a horse, it will naturally unload a leg when it's sore. It will happily put load on a little bit more steadily when it's not sore. It will stand on that leg. Its body weight simply standing on that leg is higher. So I don't know, maybe there's something, you know, naturally in their design, in their ability to do things which makes that process a little bit easier for them. I don't know. No idea.
Yeah,
Maybe.
yeah. I have thought of that. Like if they stand like all day. I don't know how long do they lay down, but like we sit down all day. So like maybe an aspect of tendon rehab for us would be to actually put load on the tendon throughout the majority of the day. I don't
Yeah.
know.
Yeah, I think they sleep for, it's like half an hour at night or something lying down. So it's, yeah,
Oh,
I don't know,
really?
maybe.
Yeah. So we kind of talked, yeah, young versus old tendons, because everyone when they get to like 30, 40, they start to see this performance decrease, and maybe some people trace it back to their tendons. But then we have that, I think they had that half-life of collagen was like 200 years or something. So then it's like, well, if is it coming from collagen degradation? I guess if you damage it, but it's like that stuff should be good for your life. Do you think this performance decrease, and I think usually it is like that 30 to 40-year-old athlete, they'll see like a significant performance decrease. If it is from the tendon, do you think that that's an age time where you start seeing this IFM getting stiffer, or is that actually something that doesn't happen until much later in life?
Yeah, no, that would be roughly my hypothesis. It's very, very hard to obviously get a spectrum and look at. We've done a spectrum in horses. And again, you're estimating, aren't you? So we went from horses of, well, right through development, and then from two, which is what you class as skeletally mature, to 28, 30, or something like that, which is a reasonably old horse. And you see you can plot a straight line through the stiffening of the IFM. give or take. So you can see that process is consistent all the way through in the horse. In humans, we've only really been able to look at old samples. We can sort of get what I'm going to call middle age because it makes me feel better as well. Optimistically called middle age in your 50s and the such like. And we can look at that and we can look at sort of 80s, 90s. We don't have access to the sort of full spectrum of tissue in the same way at the moment. So hopefully we will find out because we're trying to run the ultrasound studies at the moment. But some of the ultrasound studies that have been done would certainly look at, young is usually your students who will participate in a study I'm sure their middle age group was something very offensive like 20s and 30s, but you could certainly see a stiffening happening in the tissue there as well. So it would seem that that is not unreasonable that you're starting to see IFM stiffening if there is a point at which that just starts to reach functionally, has a functional implication. That would make sense to me.
What do you think is causing it to get stiffer?
Potentially just, sl- yeah, my best guess would be that you work every day, you use this tissue, you create a bit like you do with muscle when you load it, very small, localized, tiny amounts of damage, and that's pretty healthy because that's what the tissue goes. Oh, yeah, okay, very minor cell response. Clear out that, replace it, turn over the matrix. Um, I think that process happens day in, day out, all the time as you're using the tissue. And sometimes you use it just a little bit too much, and that you'll start to accumulate a little bit in that tissue, just a bit more damage and slightly more of a turnover. I think there's this steady accumulation, and slowly we see some changes in cell phenotype around that time in terms of you start to see the cells behaving a little bit differently, kicking out different matrix. So there's maybe some interaction between a little bit of aging of the cell, which slightly changes how it produces matrix, how responsive it is to producing matrix, combined with this slow process of turning over tissue and just pushing it a bit too far, a bit too often. And the repairability of the cells or what it lays down isn't just not quite as good as what you had before. That would be my guess.
Yeah. Yeah, it reminds me of the circadian stuff on tendons, how as you get older, the magnitude of I guess Michael Care calls it the build up and the cleanup every day, and the
Yeah.
magnitude gets worse, and then the circadian rhythm might actually extend a little bit as you get older.
Yeah.
Yeah, so just worse. So I tell people they can probably do what they had done prior. You just need more days off between, I think.
Yeah.
Yeah.
That sounds pretty sensible.
So if the IFM is getting stiffer with age, what I don't know if you were looking at this currently. How can we avoid that? Are there any methods or like therapies or?
It's $1 million question, isn't it? Um, so there's a few things I don't know. There's a few things I'm hypothesizing that we'd like to look at. One, which would be just lovely, is can we stop it with the right type of exercises, be it sort of stretching, heavy, slow or something. There's a part of me that feels like heavy, slow loading ought to allow sliding and keep it moving. I've no idea if that is true or not. So that's one thing we want to start to explore and see if it might work. The other thing I'm quite interested in is how we might be able to go in at a cell level, because I talked about this very early response to overload. It kicks off this response in the IFM cells. We get from there to spaghetti mess, and we don't quite know how. Typically, what you see in a tissue, in musculoskeletal tissues, is the cells will signal to each other. So I've been talking about IFM cells only responding at that stage. I would expect that will kick off a signaling cascade. It might initiate a response in the fascicular matrix cells, and the two between them are responding and turning over the matrix and potentially setting a cycle to damage. If that is the case, maybe we can go in with some sort of target treatments that manipulate that cell signaling. So one of the other things I've sort of got quite interested in is building in vitro models, so organ chips of tendon. So building a miniature model of tendon on an organ chip environment and using it to ask questions about when you load the cells, how they're responding, what the signaling pathways are that might IFM cells might talk to the FM cells and kick off a major inflammatory response. And if we can manipulate that or edit that or change it in a way that would stop that cycle from heading downhill.
So I don't know if you answered it there, but what's what are you working on now? What
So
do you
yeah,
think? Yeah, yeah. What are you working on now, and what do you think in the future people we're gonna be able to take from all this IFM stuff?
so that's the two things. So yes, we're working on the imaging. We're working on trying to get, so we're teamed up with Jeff Bamber, who's an ultrasound expert who developed a technique called slip elastography. He developed it actually to look at tumours, and particularly tumours in the breast, to help manage operations to see how well adhered they are to the surrounding tissue. But he's looking at sliding planes in tissue, yeah, to see if tissue slides. So we said, well, can we use that to look at sliding planes in tendon, identify where they are and how much shearing there is. And we've been building that up for a while. And we're trying to get some funding at the moment to really do some reasonably big cohort studies to look at young people, older people, injured people, looking over recovery, try to get this kind of sense of what's happening in the tissue, how it's sliding, and then perhaps track different types of treatment and see if you can what treatment approaches are helping create non-uniformity and tendon deformation. So that will be fun if we can get that up and running. So that's one side. And yeah, the other thing I spend a lot of time on really at the moment is moving in and looking a bit more at that cell level. So we've spent forever trying to actually extract and phenotype and understand what IFM cells are. It's been really difficult because we haven't got very good markers for cells. We don't know how to, extract them, work with them. They're very interesting little things. If you try to just grow them like you grow ordinary cells, they just stop growing and you lose them. So if you put all of the cells from a tendon, you just digest the tissue and plate them on plastic and grow them, within a couple of weeks, you will have nothing but the fascicular matrix cells left. All the IFM cells will just stop growing because they don't like the stiff substrate of tissue culture plastic. Well, that's reasonable, isn't it? We just said they live in this soft hydrated gel. So we're starting to work out, understand a little bit about them, understand their phenotype, how they behave, and building models so we can start to ask these questions now about if we overload these cells, how are they responding? What's being kicked off? How is that leading to damage and inflammation? And can we tune it? Should we tune it? I'm not sure we should stop it. Certainly not all of it. That might be bad. Some inflammation is probably good. It's healthy, but are there ways with treatments we can manage that? And we did try putting in injections actually as well. We had a little bit of a look at this. We could actually sort of inject lubricin, like hyaluronic acid type substances into the tissue to see if we could help with shearing. It seems quite hard to do though, and a lot of them are thicker than the actual, so they're more viscous than the IFM itself. So it's not had much luck with that at the moment. But I wonder if there's anything physically we can do with injections that might help with shearing as well.
Um, Hyaluronic acid, is that a big component of IFM or
It's in
not?
there. It's certainly
It's
more in there than it is in the fascicular matrix. Yeah, it's not the main component, but it's certainly one of the key proteoglycans there. Yeah, so
yeah.
it's interesting.
Yeah, I had seen I think it was a recent study on acute tendon when they get the swelling and they were injecting a high hyaluronidase to break down the excess hyaluronic acid. So yeah,
Mm.
it seems like there's a probably something is good, but then when it's excessive, maybe not good, you know?
Yeah,
Yeah.
often the way though, isn't it? It's
Yeah.
it's usually about balance. Yeah.
Yeah. All right, Hazel, can you tell everyone, I mean, you're not much online, but can you tell them where to find you?
Sure. Yes. So I'm at Queen Mary, University of London, and I'm in the School of Engineering and Materials Science there. So I think if you look me up straight, QMUL, you'll find me, and you're welcome to get in touch.
Great. All right, thank you for coming on.
All right. Well, thank you
Yeah.
for chat.